Thank you so much, Mike, and thank you all of you for being here this morning to share this very exciting event. We just heard from Mike about some amazing research being done here at Northeastern to fight some really terrible diseases caused by microbes. And for most of us, for most of our life, this is the context in which we've thought about microbes as something bad, right? As germs that must be eradicated or else they're going to harm us. But what's become apparent is that microbes are actually also very important to us. And indeed, they're actually a huge part of us as our microbiome. Our microbiome, you may or may not know, makes up the majority of the cells in our body. You actually have more microbial cells in you right now than human cells. And in fact, in your gut alone, there's more than 1,000 more microbial genes than you have human genes. And what we're finding, amazingly, is that the microbiome, in particular also the gut microbiome, is really essential to our health. For example, it's extremely important, essential to the proper development of our immune systems. And we're finding a lot of association between altered microbiome and a number of different diseases. And how do we have an altered microbiome? Well, there's natural alterations that occur in our microbiome with development, with aging, but there's also changes in the microbiome that can occur with exposure to different types of factors. So when we say different types of factors, one thing that we're particularly interested in our lab is the food that we eat. And when we think about an altered microbiome and the diseases that it's associated with, some of them sort of make sense, right? Because they're diseases which are associated with the gut, for example. Things like obesity, things like diabetes. But some of them are not as logical in that they tend to be associated with our central nervous system or our brain. So amazingly, we're finding links between an altered microbiome and things like Alzheimer's, depression, and even autism. Now, one of the major challenges in sort of understanding these links is a lack of proper model systems in which to study them. So most of the studies that we do of the impact of our microbiome involve rodents. So we actually take samples of human microbial cells and introduce them to rodents to look at what effects that they have. I probably don't have to tell the people in this audience there's limits in the translation, right, of response in a rodent in translating to what we would expect in a human response. So one way that we're trying to address that here at Northeastern and working with collaborators is by developing tissue-engineered gut models that capture some of the very complex biology that exists in the gut. So these tissue-engineered models sort of capture key features of what exists in the gut and governs the interactions that we have with our microbiome. So we have, for example, model microbial communities and immune cells. And we're facing some really interesting questions in developing these tissue-engineered models. So for example, we're collaborating with Kim Lewis's lab in biology to try to understand why it is that certain microbial communities that we introduce to these systems seem to activate an immune response and compromise our intestinal barrier while other ones don't. So it raises some interesting questions about, what governs that response. Is it just the types of microbes that we have in our system? Is it certain numbers of microbes? Or is it things that we're feeding those microbes and the resulting metabolic products that they're producing? So we're also collaborating with other labs at Northeastern, for example, the Kappa's lab in chemical engineering, to introduce neuronal cells into our system, which are really important for interactions between the microbiome and the gut and other organs in the body, including the brain. And importantly, we're collaborating with labs like the Golick lab in chemical engineering and also the Griffith lab over at MIT to develop fluidic culture platforms that allow us to introduce our model gut and also other model organ systems, including a model organ brain, so that we can start to understand some of these interactions between the gut microbiome and different types of diseases involving the brain. So one thing that we're doing right now is we're using this tissue-engineered model gut system to start to explore some of the interactions between foods and food products, the microbiome, and health. So you heard from Jacqueline Locke, a wonderful graduate student that I'm super lucky enough to have in my lab this morning, that she studies intestinal mucus. And one interesting finding that Jacqueline has had is that it appears that certain food additives, common food additives like emulsifiers, which are present commonly in processed foods, appear to change the ability of microbes to swim through intestinal mucus and penetrate this very important barrier that exists in our gut between the microbiome and underlying tissue. And this might be part of a link to understand interaction or links that have been made between food additives in processed food, metabolic syndrome, and obesity. We've also found that other food products appear to strengthen the mucus barrier, and we're now trying to understand, can we exploit these effects, for example, for therapeutic purposes to prevent infection? We're working in collaboration, as has been mentioned in the other talks this morning, with a number of people not only at Northeastern University, but leveraging the rich research environment in Boston. And we're starting to try to use our model gut system for trying to understand these interactions with our microbiome, with the goal ultimately of understanding, can we control the interactions between our microbiome to benefit human health? Thank you.